Good morning, students. Myself, Dr. K. Sivaya, Department of Pharmacology, TVM College of Pharmacy. So, one more topic is present in second unit, that is carbohydrate metabolism. So, regarding that, how the blood glucose regulated in our body is very important. If you observe the glucose in our body, how it will get. So, the sources of glucose mainly diet. So what you had food, that diet have the carbohydrate, that carbohydrate break down into glucose molecules. That is one source. And gluconeogenesis. This gluconeogenesis takes place in the liver. Then glycogenolysis. Breakdown of glycogen molecule into the glucose molecules. Already we studied, I uploaded video regarding glycogenolysis. These are the three sources of the glucose molecules. Fine. Then how the glucose is utilized in our body. So the glucose first, after taken by the cell, the glycolysis takes place for the energy production. Then after that, Krebs cycle or TCS cycle. Then one more, if the alternative pathway is exose monophosphate, but if the glycolysis not takes place, TCA takes not happen, the HMP exose monophosphate pathway happen. Then glycogenesis. If the excess of glucose molecules, if it is available, if it is available in the body, the glucose is converted into glycogen. That is, we can call it as glycogenesis. Or it is converted into lipid. That's why it is called as lipogenesis. If it is excess availability of the glucose molecule, it is either form the glycogen or a lipid. This is the how the body is utilized in our body, and the sources of glucose we discussed. Fine. Come to the blood glucose level in bloodstream. Blood glucose level in the quantity. In the fasting, means night, if you 12 hours, if you fasted, in the early morning, if you went, if you taken the blood glucose in the plasma, if you taken, it will be 70 to 110 mg per deciliter. And post glucose level, this is post glucose level, nothing but after eating food, after 2 hours, because of 2 hours take the time for the absorption of these carbohydrate and metabolism. So after to post glucose level, that is 110 to 140 mg per deciliter. And random means we can take any time, either in the early morning, either fasted, or if you taken food, before food, if you taken, it may be 70 to 140 mg per deciliter. These are the ranges of blood glucose levels in the bloodstream in normal person. If it is crossed, nothing but 160, 180, 200, 250, 300 mg per deciliter, if it is there, we can consider it as diabetes mellitus. We can consider it as diabetes mellitus. So the glycosuria, the glucose present in urea is called as glycosuria. So here the 180 mg per deciliter, if we can term the glycosuria when the blood glucose levels more than the 180 mg per deciliter in urine, we can call it as glycosuria. Fine. You already heard about hyperglycemia. So when the glucose levels increases in the body, we can call it as hypoglycemia, hyperglycemia. Nothing but more than the 150 mg per deciliter, 160 or 180, 200, 250. If it is there, we can consider it as hyperglycemia. What are the symptoms of these hyperglycemia? Frequent urination. If you observe, the diabetes patients are more going to their urination come continuously, maybe per day, 8 times, 10 times. Then high levels of sugar in urine. So high levels of sugar. Thirsty feeling frequently. They always thirsty feeling because of they are getting frequently urination. That's why they need more water. High blood glucose and causes. So what are the causes for these hyperglycemia? Maybe insulin not secreted properly. The insulin secreted with beta cells of the pancreas, or sometimes less consumption of insulin. So the patient taking the very less insulin, or if the patient taking the insulin but it is not utilized by the body, and overeating, taking of overfood, lack of exercise, stress from an illness. The stress also one of the causing agent to getting the hyperglycemia. Stress over personal problems. Okay, these are the causes of the developing of hyperglycemia. And solution: exercise frequently, cut down the amount of overeating food, take insulin as prescribed by the doctor. So whatever the doctor prescribed in the prescribed manner, okay, and dose is essential to take the patient. Check your blood glucose more often. Every if the diabetes patient says every 10 days they have to check blood glucose levels. And what is a hypoglycemia? Hypo means decreases glycemia's blood glucose level. The decreases the blood glucose levels we can call it as hypoglycemia, or decreases the low levels of glucose, we can call it the low levels of glucose we can call it as so that is less than 70 mg per deciliter. What are the symptoms of insulin? Sweating, shaking, and fast heartbeat, dizziness, anxiety, nausea, and sometimes vomiting also, frequent headache, fatigue, anger, and unconsciousness. These are all the symptoms of the uh, hypoglycemia. Means the patient have the don't have energy. If you just you feel once, if you don't have energy, whatever in our mind, whatever your behavior, that all behaviors comes under hypoglycemia. So happen when it is body produces too much insulin. So when it does happen, when the body is too much insulin, because of insulin always reduces the decreases the blood glucose levels in body. Okay, and solution: recheck your blood glucose regularly and consume about 20 grams of glucose. So if the patient, if the hypoglycemia, if the patient is hypoglycemic patient, immediately we have to give the some glucose. Not exact 20 grams, we can give any up to patient getting the consciousness. So we can use glucose tablets, gumdrops, or hard candies, and it's glucagon kits. Why it is glucagon here? The glucagon increases the blood glucose levels. So that's why we have to give the glucagon. We can discuss later what is the glucagon function. Fine. Up to here, what you studied, what is the sources of the glucose, and how it is utility, and what is hyperglycemia, what is hypoglycemia, and symptoms and causes. Now come to the main topic is hormones involved in regulation of blood glucose. Already we studied how the glucagon is regulated and insulin regulated the blood glucose levels in the body. The other hormones also to regulate the blood glucose is epinephrine. It is secreted from the adrenal medulla, and cortisol, it is a glucocorticoid. It is secreted from the adrenal cortex, and other hormones, growth hormone, and adrenocorticotropic hormone, and thyroxine. These are all involved in the regulation of blood glucose levels in the body. So in the recording, so which hormone to decreases the blood glucose level is insulin and somatostatin, but 80 to 90%, 80 to 90% of the blood glucose levels decreased by insulin, right? Insulin. And which hormone increases the blood glucose levels? Mainly glucagon. 80 to 90% the blood glucose levels increased by glucagon. Other hormones are epinephrine or adrenaline. Epinephrine is also called as adrenaline. And cortisol, that is glucocorticoid, adrenocorticotropic hormone, and growth hormones, and thyroxine. These are all hormones increases the blood glucose levels in the body. Dot. Usually these insulin and glucagon, where it is secreted, already maybe you know, it is secreted from the pancreas. These three hormones are very important and regulate the blood glucose levels in the body. The glucagon is secreted from the alpha cells of the pancreas. Okay, and beta cells secretes the insulin, and delta cell secretes the somatostatin, which regulates the alpha and beta cells. Somatostatin regulates the alpha and beta cells. Okay, fine. Insulin. It is a peptide hormone synthesized or secreted from the beta islets of the pancreas. It regulates the metabolism of carbohydrate and fats, promoting the absorption of glucose from the blood to skeletal muscles and fatty tissues. So here the insulin function, the function is to engulf the glucose molecule into the cell. Means we can discuss that mechanism is there. Metabolic effect enhance the entry of glucose into the cell, and that glucose stored as glycogen or conversion of fatty acid, enhance the synthesis of fatty acids and proteins, suppress the breakdown of protein into amino acids and adipose tissue into free acids. So glucose molecule, if it is there, there is no, it suppress the breakdown of proteins or amino acids or adipose tissue. See, now this is the main mechanism action of the blood, main mechanism action of the insulin. See, blood when the blood glucose levels increased in body, when the see when the blood glucose levels increase in our body, insulin is released. Okay, these insulin bind to the cells of the especially skeletal muscle and smooth muscles, and it is bind to the receptors. So the insulin binded here. Then endocytosis, not exocytosis, it is given wrong actually. It is endocytosis of glucose molecules. So whatever you had put, that food particles, this is glucose molecule. Just to observe my arrow mark, this glucose molecule, this glucose molecule, this is insulin. This is insulin secreted from pancreas. So these insulin binded cell membrane of the insulin receptor, here the glucose molecules are entered into the cell. The insulin, if it is binded to the cell membrane of the cell, then only the glucose molecule entered, otherwise not entered. It is a like insulin is a like one lock. If it is insulin, if it is key open, the glucose molecule entered, otherwise not. That is the main mechanism of the insulin. If insulin not, if it is not there, the glucose molecules not enter in our body, nothing but not enter into the cell. That's the thing. And one more. After that, the glucose molecules, if it is more, it is converted into glycogen in the cell. Sometimes it also activation of glycogen synthase. These insulin activate the glycogen synthase enzyme. Already we studied in glycogenesis, synthesis of glycogen. Already we studied, already uploaded the video, glycogen formation. For that, it is required glycogen synthase enzyme. So these insulin enhance the or activation, increase the activation of glycogen synthesis, and because of that, the glycogen formation increased. Okay, because of these two functions, the glycogen more formed in the skeletal muscles, smooth muscles. This is the mechanism of the insulin. And one more is there, we can discuss later. After discussing about the glucagon mechanism of action, one more mechanism is there, insulin, we can discuss. Somatostatin, just this is growth hormone and inhibiting hormone. Why it is called? It inhibit the or suppress the both alpha cells, suppress the glucagon release from alpha cells, and suppress the release of insulin and pituitary hormones, gas and secreting. But it also it enhances some mechanisms. That's why it is called growth hormone. Okay, fine. This is somatostatin. But we have to remember that somatostatin suppress the glucagon release from the alpha cells and suppress the release of insulin from beta islets. Next, glucagon is one more important. It is also peptide hormone secreted by the alpha islets of the pancreas. It raises the glucose levels in the body. It is exact opposite to the insulin. Okay, it decreases the glucose levels in the blood. Say in metabolic effect, enhance the release of glucose from the glycose. It enhances the release of glucose from glycogen, enhances the synthesis of glucose from amino acids or fatty acids. So this is the main mechanism of the glucose gun. Increases the glucose levels in the body. Insulin is decreases the blood glucose levels in the body. So action of glucagon, the main mechanism action of the glucagon. When the blood glucose levels, decrease in our body, the glucagon released from the alpha islets. The glucagon released from the alpha islets. So these glucagon bind to the cell membrane of the cell and activate the phospholipase enzyme. When the phospholipase enzyme activated, this phospholipase enzyme releases the adenyl cyclase. That adenyl cyclase enzyme act on the adenosine triphosphate. These adenosine triphosphate is converted into ADP, adenosine diphosphate and cyclic AMP. These cyclic AMP, this is activation of adenyl cyclase, conversion of cyclic AMP. When the cyclic AMP levels increase in our body, the function is activation glycogen phosphorylase. In the glycogenesis, already I uploaded a video. In that glycogen formation, that is enzyme is a glycogen phosphorylase. When the glycogen phosphorylase enzyme is activated, glycogen phosphorylase is activated, glycogenolysis, sorry, not glycogenesis, glycogenolysis. I'm very sorry. This is the glycogen phosphorylase enzyme present in the glycogenolysis enzyme. The glycogen molecule degraded into glucose molecules. Just you go through once glycogenolysis. Because of that, release of glucose into the blood. And one more function of this cyclic AMP is inhibition of glycogen synthesis. Inhibition of glycogen synthesis. You know, if the glycogen synthesis enzyme is inhibited, there is no formation of glycogen from glucose. So the glucose molecules as it is, there is no formation of glycogen. So the increases the blood glucose levels in the body. Next, insulin mechanism of action. When the blood glucose levels decrease in our body, the insulin is released into the bloodstream. That insulin it's bind to the cell membrane of the cell. Then after that, it utilizes the cyclic KMP and to convert the 5 adenosine monophosphate in the presence of phosphodiesterase enzyme. Once I am repeating, when the blood glucose levels increased in our body, the insulin is released from the beta cells. That insulin bind to the membrane receptor of the cell. After that, what is doing? Whatever the formed cyclic KMP, it convert into the AMP, adenosine monophosphate, in the presence of phosphodiesterase enzyme. Because of this, there is no formation of, just to observe here, there is no formation of cyclic KMP. So there is no activation of glycogen phosphorylase. There is no glycogen phosphorylase. There is no formation of blood glucose. So there is no degradation of glycogen into glucose. So when the cyclic AMP levels, here it is not formed. The cyclic AMP it is formed automatically. This process is entire different, opposite. Means blood glucose levels decreases. Here release blood glucose, but it is not released because of there is no activation of glycogen phosphorylase and inhibition of glyco. Here it is in here now it is glycogen. It is stopped. The inhibition so that glycogen synthetase enzyme as it is worked. If the glycogen synthetase as it is worked, the glucose molecule is converted into glycogen. That is the mechanism action of the. It is deep in the cellular level. It is an insulin mechanism of action. So overall, if you observe, low blood glucose, if the blood glucose levels decreases, stimulates the alpha islets and releases the glucagon. That glucagon stimulates the especially liver cells and enhance the glucose in the body. That is called as gluconeogenesis. Already we said gluconeogenesis maximum takes place in liver. So the glucagon stimulates the liver cells and enhance the release of glucose from other pyruvate, lactate. So the release of released and come to the normal. Then if the blood glucose levels more, since more glucose in the blood, what happened? So the pancreas releases the insulin from the beta cells. These insulin enhance the, already I said, engulf the glucose molecules. Because of the insulin enhance the absorption of glucose into the cell. Then there it is a formation of glycogen and fat, lipid. So then come to the normal. So these are the main important two mechanisms you have to write in the examination. The other hormones also involved, that is epinephrine and epinephrine. See, enhance the release of glucose from the glycogen, enhance the release of fatty acids from the adipose tissue. Because of this, epinephrine raises the blood glucose levels. Already I said in one slide, except insulin, the remaining whatever we discussed, the hormones, max, all are increases the blood glucose levels. So enhance the release of glucose, enhance the release of fat from adipose tissue. So when the epinephrine or adrenaline released into the bloodstream, it acts on the liver cells and produces the glucose molecules from glycose and amino acids, other substance glycerol. So glucose molecules are produced. Cortisol. Enhance the gluconeogenesis. same. How the epinephrine is act, same. And antagonize the insulin. This is one more activity of cortisol. Antagonize the insulin. Insulin function opposite. So antagonism means to inhibit the insulin function. So because of this, glucose levels rises. This is the how the cortisol released into the body. This is it will come after in the in our human anatomy physiology. Other hormones, adrenocorticotropic hormone and growth hormones also enhance the glucose levels. So enhance these adrenocorticotropic hormone release of the cortisol and enhance the release of fatty acids from adipose tissue. Because of this rises the blood glucose levels. So how these adrenocorticotropic hormone act, increases the cortisol and increases the blood glucose levels. And growth hormone, it mainly antagonizes the insulin. Growth hormone is to antagonize the insulin function. This release in anti-inflammatory. Because of this release rise of blood glucose levels. And thyroxine. Thyroxine increases the metabolism, body metabolism. So because of this, enhance the release of glucose from glycogen. So glycogenolysis. The thyroxine function is glycogenolysis and enhances the absorption of sugars from intestine. The more absorption, more glucose levels are present. Because of this, rises the blood glucose levels. So this is the how the blood glucose levels regulated in our body. The main blood glucose levels regulated by insulin and glucagon. Remaining others are also maintained and regulated, but these two are very important for writing in the examination point. Thank you.